And what does mercury do to the heart? A published article out of the University of Rome took 13 cases of cardiomyopathy, like me, with left ventricular dysfunction, low ejection fractions, awaiting transplant, basically. They took 24 cases of other heart disease of other type and 4 controls, and they biopsied the endomyocardium and took out little pieces of heart tissue from the right ventricle. And they took this tissue from all these patients and put it in a nuclear reactor in Rome and radiated it with a neutron flux, which makes all the heavy metals in that tissue radioactive, because they absorb neutrons and become radioactive. Once radioactive, they secrete gamma radiation at a certain spectral frequency, allowing them to accurately quantitate the amount of mercury in the tissue to the atom. They could determine how many atoms were in a piece of tissue. What they found was striking. All 13 cases of cardiomyopathy had 22,000 times more mercury than normal. 22,000 times more. All 13 of them, compared to the group of normals. And something like 12,000 to 10,000 more than the other heart disease, coronary disease, valvular disease, etc. A professor, brilliant professor from University of Kentucky, did a calculation as to what it would take to get 22,000 times more mercury into a heart muscle cell. And he concluded that there weren't enough binding sites for that much mercury unless there was some other problem going on. In other words, what he's trying to say is that I'm not sure so much that mercury actually drives this so much as it is you become a mercury canary and just sort of collect it in the cells that have inadequate glutathione defense. So sort of it's a player, but probably not a primary player. But this is what I mean by the xenobiotic toxicity factors that follow and seem to generate what we call the classic syndrome of chronic fatigue syndrome. My brain's going, I have pain, and I have no energy. And the xenobiotics, xeno means foreign, biotic means molecule. Foreign molecules not of humankind. From the gut, they are the waste products of 2 billion bacteria. They're even normal bacteria, but they do have waste products. And these waste products are highly toxic. And they might be in your root canal teeth or your dead teeth or both. But if I had to guess, what am I going to be more worried about? 2 billion bacteria in your colon or 10 million in your root canal tooth? Yeah, I'd be a whole lot more worried about your colon. So people, I do not advocate that people with root canal teeth go out and yank their root canal teeth and yet leave their colon. The result of that is that the incidence of significant improvement by yanking the root canal teeth is not very great over leaving them in, because you mistake the problem. The problem really is a global xenobiotic toxicity problem, and not just the root canal is just one of many things. Environmental toxins, of course, could be involved, and a lot of people. I mean, you could point your finger at millions and millions of xenobiotic problems. The basic problem is if you don't have enough energy, you don't have defense against any of those things, but most especially the one that is most common, and it's below your navel. Then comes the microcirculatory issue, or cardiomyopathy. And boy, when that gets into the picture, especially as you defend against it with your adrenergic tone, significant problems can then accrue, in which these things begin to interact. Indeed, if you don't have circulation, you don't have decent immunity. If you don't have decent immune surveillance, things start activating, and pretty soon you get another virus coming up on the heels of this thing, and then you're back to number one. Or if cardiomyopathy causes a leak in the gut, then you get more of number two. So they all are interacting. So it's hard to figure out what's the chicken and the egg here. It's a big soupy mix. last but not least, is your genes expression changes. Susan Vernon at the CDC has identified at least 8 genes that are expressing themselves either greater than or less than normal. I suspect that there's a lot more than that. You're seeing the genetic phenotypic expression of both the disease itself, I suppose, but more importantly, your genetic defense against it. This genetic phenotype shift can seat your disease at the genetic level, and the things causing it, if they went away, you would never know it, because now you're operating as if you are sick at the gene level. And they interact with each other, and there are consequences to loss of energy. One is the more energy you lose, the more acidotic you become. You know, as you lose mitochondrial energy, you upregulate your non-energy producing pathway, and it makes lactic acid and other organic acids. So you get more and more acidified intracellularly. You respond initially by an extracellular alkalosis, which causes vasoconstriction, making microcirculation even worse, especially in a low cardiac output state. Extracellular alkalosis, by constricting vessels, can impede oxygen transport, so that when I have you hold your breath with the pulse oximeter on, you don't desaturate. Why? You have no microcirculation to desaturate with. As you lose glutathione because of energy problems, you can't convert citrate to isocitrate, so citrate rises in the blood, and we've measured it in the urine, it's very high. And citrate is the most potent inhibitor of 2,3-DPG known to man, and as it inhibits 2,3-DPG, you can't even take the oxygen off the molecule called hemoglobin. So this is a livid mess. It just gets messier. What surprises me most about this is that you survive this. Not that you don't have anything going on, that you would survive something like this. It's just remarkable. And we have a clue as to why you do survive this. When you have extracellular alkalosis, you get volume contraction. The worst thing you could possibly have with a diastolic dysfunction is volume contraction associated with it. So what the cardiologists are missing is it's one thing to have diastolic dysfunction without volume contraction, but it is another thing still to have diastolic dysfunction with volume contraction. When you have diastolic dysfunction and you don't fill well, it's important to have sufficient filling pressure, and you can't get that way if you have a reduction in blood volume. You also begin to spit out magnesium, because citrate is a big binder of magnesium, so magnesium losses accelerate to the point that the body becomes significantly magnesium deficient. When that happens, calcium can no longer be inhibited from entering the mitochondria. It's inhibited by calcium, by magnesium, and the mitochondria become what is called calcium. It's called calciphylaxis of the mitochondria. It basically brings down the mitochondrial function. So this system is replete with positive feedback loops. As the system gets worse, those things that get worse make the whole thing worse, and around you go. And by the way, this type of magnesium deficiency cannot be corrected. A study out of Belgium by Greta Morkens showed that something like 60% of patients were body-wide depleted of magnesium with normal serum magnesium levels. Because you remember, serum levels are determined by kidney function, but intracellular levels can be low with a normal serum value. And she said that no matter how much magnesium she poured into these people, IV, etc., only could correct 40% of those with deficiencies, because without energy, the magnesium leaks out against its gradient. You can't maintain intracellular magnesium. This is the pulse oximetry data. This is what we did was we just had people with a pulse oximeter on their finger, and I had them hold their breath for as long as they could, and you can see how long they held their breath on the y-axis. 10 seconds, 20 seconds, 30 seconds, 40 seconds, 50, 60. One guy made it to 80 seconds holding his breath before he gave up the ghost. And off to the x-axis is the percent desaturation, the change that's supposed to occur when you hold your breath. You should drop as you As you hold your breath, you build up CO2. CO2 changes the hemoglobin dissociation curve, and eventually start offloading oxygen, and the saturation begins to fall. Normal is about 6 or 7 percentage points down. So I only had, in this study of 60 patients, only 10% of them were normal. More interesting than that, look at about half of them at 0 to 1. That's 30 or 50%. 50% of the patients failed to desaturate 1 point following a maximum breath hold. One guy went 80 seconds saturated. 1 point, by the way, is the variance of the test. So it's, so 1 could be 0, or 0 could be 1.